everyone, welcome back to another video of STEMAC, and today we're going to learn about DNA and RNA structure. So first let's talk about DNA. So before that, in our previous nucleotide video we talked about what nucleotides are, right? So this is basically a nucleotide where we have a pentose sugar and a nitrogenous base, and at the other side a phosphate group. So DNA and RNA both are made up of nucleotides. So when we bring multiple nucleotides together, we get polynucleotide, and DNA and RNA both are polynucleotides. So how are polynucleotides formed? We learned that when we put two nucleotides together, they at the third carbon we have a hydroxyl of the pentose sugar, and the pentose sugar forms a phosphodiester bond with the phosphate group of the next nucleotide, right? So that is how a polynucleotide is formed. So this we see here, this section is a polynucleotide. So here we have one nucleotide. This is the pentose sugar, this is the base, this is the phosphate group, and at the third carbon the hydroxyl group has formed a phosphodiester bond with the phosphate group of the next nucleotide. So when there are multiple nucleotides together, we get a polynucleotide. Now in the case of DNA, we need two polynucleotide strands. Therefore DNA is a double helix structure. So here we can see that there is one polynucleotide strand and here is another polynucleotide strand. So this is actually the backbone, the sugar and phosphate, sugar phosphate backbone, and at the middle we have the bases, nitrogenous bases. So when we before that we need to know that the nitrogenous bases of DNA there are four types, right? So they are adenine, thymine, guanine, and cytosine. And we learned that adenine and guanine have two ring structures, double ring structures, and thymine and cytosine have single ring structure, right? So these are, as we know, purine, and these two are pyrimidine, right? So during in the structure of DNA, the double helix structure, what happens is that the these bases are complementary to each other. So when we get we have an adenine base in this section, right? So for this adenine we will need a complementary thymine. So adenine and thymine are complementary together, while guanine and cytosine are complementary to each other. So adenine and thymine, when we have an adenine we will need a thymine. When we have a cytosine we will need a guanine at the other strand of the nucleotide, polynucleotide strand. Again, when we have a adenine we get a thymine, and it goes on and on, right? So we need to know that how these two pairs up together, these two strands, nucleotide strands, polynucleotide strands. So we get they are bound together with hydrogen bonds. Now there's a slight difference between the adenine thymine pair and guanine cytosine pair. So in case of adenine and thymine, when they form a hydrogen bond, they consist of two hydrogen bonds. So there are going to be two hydrogen bonds in case of adenine and thymine, and in case of guanine and cytosine we will get three hydrogen bonds. So it's 3 in case of guanine and cytosine. So we can tell that to form a double helix structure we will need a purine and a complementary pyrimidine. So for adenine is a purine, right? And its complementary is thymine, which is a pyrimidine. And in case of guanine, which is a purine, we will need a complementary cytosine, which is a pyrimidine. So a for a purine, we will need a pyrimidine base. So this is basically how we get the double helix structure. So as we can see here, these are the hydrogen bonds, and if this is adenine and this is thymine, then we have 2 hydrogen bonds, and if this is guanine, this is cytosine and this is guanine, then we have 3 hydrogen bonds. So now we need to know about another interesting fact, and that is that the strands, the 2 polynucleotide strands of DNA, they are anti-parallel to each other. So how is that? Here we can see that this phosphate group, we know that the phosphate group is bound to the 5th carbon of the pentose sugar. This is the 5th carbon of the pentose sugar. So in this strand, the phosphate group is at the top, right? So this is the 5' end of this strand, because the 5th carbon is at the top and the phosphate group is bound to that 5th carbon. And at the bottom we can see that the 3rd carbon, where the hydroxyl group is present, it is at the bottom. So this is the 3rd prime or 3' end of the polynucleotide strand. Now if you go to the opposite strand, we can see that the 3rd carbon is now at the top and the 5th carbon, where the phosphate group is attached, is at the bottom. So they are anti-parallel to each other. And as I said, the purine and pyrimidine will always bind to each other, so they are complementary. Now that it has a reason why that is complementary to each other. So if we let's say we had to attach adenine and guanine, I mean the 2 purine together, let's say, what will happen in that case is that we need to the 2 polynucleotide strands of the DNA have to maintain a certain distance, or they have to be at a certain width. So if we were to put the purines together, they would have required a distance of more than 2 nanometers. But if we were to put these 2 pyrimidines together, the thymine and cytosine together, these 2 pyrimidines, they would have required a distance lower than 2 nanometer. So in this case, the distances are not similar, right? If we were to put adenine and guanine together, we would require a distance more than 2 nanometer, and in case of thymine and cytosine, we would require distance less than 2 nanometer. So it will not be a, it will hinder the formation of the double helix of DNA. So that is why when we put complementary bases together, which is adenine, thymine and guanine and cytosine, we get 2 nanometer width between the 2 polynucleotide strands. So they are always going to be at a width of 2 nanometers, and this is why it's required to put pyrimidine with purine. So thymine and adenine and guanine and cytosine. Now because of this conformation, the hydrogen bond between the adenine, the purine bases and the pyrimidine bases, we get a coiled structure of DNA, right? So this is actually what a DNA looks like. This is, if this is one strand of the DNA, double helix structure of DNA, and this is the 5' end, then we get a 3' end at the bottom. And if this is the second strand, polynuclear strand of the DNA, then if it's going to be anti-parallel, so this is going to be the 3' end and this is going to be the 5' end. So this is basically what a DNA, actually basic structure of a DNA. Now if we move to RNA, we learned that RNA is also made up of nucleotide. Now there are some key differences between DNA and RNA, which is we learned previously in the nucleotide, our nucleotide video, which is that the sugar that we use for the DNA nucleotide and the RNA nucleotide are different. For DNA it is deoxyribose, and for RNA it is ribose. And in ribose, the second carbon has a oxygen, while the deoxyribose sugar have the lack of an oxygen molecule. So when in case of DNA, the main difference is that in case of RNA, the main difference is that in DNA we require 2 polynucleotide strands, where with RNA we just need a single polynucleotide strand, right? Because RNA is also comparatively shorter than the DNA polynucleotide strand. Now, in case of RNA, we know there's another difference, which is in the nitrogenous bases. So these 4 are the nitrogenous bases of DNA, right? Adenine, thymine, guanine, and cytosine. But we know that thymine in case of RNA is not present. Instead, we have uracil, right? So similarly, the polynucleotide strand of DNA, similar to that, the phosphate third carbon of the pentose sugar, if this is the third carbon, the hydroxyl group present at the third carbon will form a phosphodiester bond with the phosphate group of the next nucleotide, and this will continue on. Okay, so let's say this is a base for adenine, this is a base for guanine, this is for cytosine, and this is for uracil instead of thymine, right? Because this is RNA. So they will form a bond, like the third carbon will form a bond with the phosphate group of the next nucleotide, and this nucleotide would form a phosphodiester bond with the next nucleotide, and this will continue on, and as a result we will get an RNA strand, which is a single strand, polynucleotide strand, unlike DNA. Now, this structure of RNA is basically very simple compared to DNA, and now we will see that there are some different types of RNA present, which are, the first one is transfer RNA or tRNA, and it's a very essential type of RNA because tRNA contains anticodons, and what this anticodons does is that this tRNA, the anticodons of the tRNA binds to the codons of the complementary codon of the mRNA, right? And when they bind to the codons of the mRNA, they bring amino acid, and these amino acids then are translated to protein. So they are really essential in protein synthesis in our body. The ribosomal RNA is another type of RNA, and they are a very essential structure or structural component of a ribosome, and ribosome also helps or also takes part in a protein synthesis, which we learned in our nucleus structure when we saw that the ribosome, ribosome carries another type of RNA is ribosomal RNA, which is a very important structural component of ribosome. Now, in our nucleus structure video, we learned that ribosomes are attached to the endoplasmic reticulum outside of the nuclear membrane, right? So these ribosomes carry mRNA within that endoplasmic reticulum, and they help in protein synthesis. So the ribosomal RNA is a part of those ribosomes, right? A structural component. And then we have messenger RNA, which are mRNA, and this mRNA contains, as we said, tRNA have anticodon, and this mRNA contains the complementary codons where this tRNA bind, and they bring amino acids, and when the tRNA binds to the mRNA with the codons of mRNA, they bring amino acid, and within the ribosome they complete the protein translation process where proteins are synthesized from amino acids. So this is basically what RNA is and the structure of RNA and the structure of DNA, and with that our video comes to an end. So thank you for watching and stay tuned for the next video.